Hi everyone. So today I'm going to talk about 11.2, which is tools of recombinant DNA technology. So this is part 1 of the video. What are the tools of recombinant DNA technology? It includes enzymes, cloning vectors, and competent host organisms. So in this video, I'm going to talk about enzymes, especially restriction enzymes. Restriction enzymes are also referred to as molecular scissors because they act like scissors. They cut the DNA at specific locations. Now, restriction nucleases or restriction enzymes are of two types: restriction endonuclease, restriction exonuclease. What is the difference between them? Restriction endonuclease will cut the DNA at specific regions within the DNA. Within. And restriction endonucleases, it will cut the DNA at the end of the DNA region. So basically from 5 prime to 3 prime region, it will cut at the 5 prime end or it will cut at the 3 prime end. So it will cut at the ends. Restriction exonuclease cuts at the end. Restriction endonuclease cuts within the DNA region. So exonucleases cut DNA at the ends. An endonuclease makes cuts at specific position within the DNA. The first restriction endonuclease is Hint2. Hint2 cuts DNA molecule at a particular point, recognizing a specific sequence of the 6 base pairs known as the recognition sequence for Hint2. What is this recognition sequence? For example, if we take an enzyme called as EcoR1, this is a restriction enzyme. It actually recognizes a sequence. It recognizes a sequence, for example, GAA, TTC, and CTTAAC. Now, before moving on to this, let me explain what is GATTC. So, A is adenine, which actually binds with thymine. G is actually guanine, which actually binds with cytosine. So adenine will always bind with thymine, thymine will always bind with adenine, guanine will always bind with cytosine, cytosine will bind with guanine. So G is C. For A it is T. Again A, T. For T it is A. So this is a whatever the opposite strand will be, G for G will be C, and opposite nucleotide for adenine will be thymine. So these are referred to as nucleotides. For example, this is a particular recognition sequence for EcoR1. Now we have a sequence, for example, it will be G, T, A, just a random sequence, C, A, G, A, T, T, C. And then the opposite sequence will be, for G it will be C, T, A, T, C, G, A, T, G, C, A, T, A, T, T, A, T, A, and G. Now what will EcoR1 do is, it will actually go through this direction and it will actually scan through the nucleotides to see which is its recognition sequence. So it will go in this direction and it will see that this is the particular recognition sequence. So here it has got the recognition sequence and it will actually make cuts within the regions of the DNA. So how do the naming and the nomenclature of restriction enzymes happen? So for example, EcoR1 is an enzyme which I have already discussed about. It is actually isolated from a bacteria which is an Escherichia coli. Escherichia coli. So how does this name take place? The first letter of the genus is here, and this next two letters of the species is here. And R is the name of the strain, and 1 is the Roman number 1, which is indicates the order in which the enzymes were isolated from the strain of bacteria. The mechanism of action of restriction endonuclease. It will cut within the DNA region. So EcoR1 is this thing. This is the recognition sequence. This particular yellow part is referred to, this is the recognition sequence. So it will cut at this end and at this end. And this is the cut part, okay, of EcoR1, and this is referred to as the overhangs. So these are overhangs, and these are referred to as sticky ends. They are sticky ends. These are overhangs. Okay. Now, each restriction endonuclease functions by inspecting or scanning the length of the DNA sequence. It will actually go, it will scan the DNA sequence from here to here and look for the restriction recognition sequence. Once it finds a specific recognition sequence, it will bind to the DNA and cut at each of the two strands of the double helix. This is one strand, this is another strand, and together they are referred to as double helix. So the two strands of the double helix at specific points in the sugar phosphate backbones. Each restriction in endonuclease recognizes a specific palindromic nucleotide sequences in the DNA. We look into palindromic. What is the meaning of palindromic? So palindromic, for example, if there is a word called as Malayalam, if you see from backwards, it is also spelled the same. Mal, So it is spelled the same backwards. So that is referred to as palindromic sequences. These are a group of letters that form the same words when read both forward and backward, as against a word palindrome where the same word is read in both directions. The palindrome in DNA is a sequence of base pairs that read same on the two strands when the orientation of reading is kept the same. For example, the following sequences read the same on the two strands from 5' to 3' direction. For example, if we see from 5' to 3' direction, it is GAATTC. Again, here if you see from 5' to 3' direction, it will be GAATTC. If we read it in the 3' to 5' direction, it will be same. If the orientation of the reading is kept the same, the letters will be same. For example, 3' to 5' it is CTTAAG. Again, from 3' to 5' it will be CTTAAG. So these are referred to as palindromic sequences. Sticky ends and blunt ends. So EcoR1 actually results in something called as sticky ends. So it will actually cut the DNA within the region somewhere here and here. So it will lead to overhangs. These are referred to as overhangs, and these are referred to as sticky ends as well. Blunt ends are somewhat, there is a restriction enzyme called SMA1, which actually cuts with a specific location somewhere here, and it will produce blunt ends. So sticky ends is easy to ligate or glued together. It's easy to ligate them, but blunt ends is difficult to ligate them. Action of restriction enzyme. So we have vector DNA here and we have a foreign DNA. So it will, EcoR1 will actually lead to what? Sticky ends. So it will lead to sticky ends, and then they'll join together with the help of an enzyme called as DNA ligase. And this is the recombinant DNA, which has fragments from from the vector DNA as well as from the foreign DNA. So restriction enzymes cut the strand of a DNA a little away from the center of the palindrome sites. So here it is cutting away from the center. It's cutting somewhere here, it's cutting somewhere here. This leaves single-stranded portions of the ends. There are overhanging stretches called as the sticky ends on each strand. This stickiness of the ends facilitates the action of the DNA ligase. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and these can be joined together using DNA ligase. So in this case, EcoR1 is used to cut this particular DNA and also this particular DNA. So the same restriction enzyme is used to cut both the strands of the DNA, and it will be easy to ligate these sticky ends because the same restriction enzyme is used. Action of DNA ligase. So we have got sticky ends here because of the process of restriction enzyme, particularly by EcoR1 in this case. And then ligation is done by DNA ligase, and it is very easy to ligate sticky ends. So so A comes here, this A comes here, T comes here, this T comes there. So this is how the recombinant DNA is produced. Action of restriction enzyme and DNA ligase. So we have a foreign DNA here and we have a vector DNA, which is a plasmid. So we use a restriction enzyme in this case. We actually produce cuts here, and there are 3 DNA fragments here, and here we have one DNA fragment. Now ligase is actually used to bind the DNA fragment into the plasmid, into the cut plasmid. Then it is introduced into host cells, particularly E. coli, and then the cells actually divide. Now we'll look into the restriction enzyme part is over. Now we look into the gel electrophoresis. How to obtain the DNA fragments from this case. This is how the gel electrophoresis actually looks like. Here there is a gel. Okay, it's not visible, but there's a gel here, and there are electrodes, anode and cathode. So the DNA here is introduced into these wells. When it is introduced into these wells, what really happens is we connect it to the electrodes, and from the DNA is actually negatively charged. So DNA is negatively charged, and it moves from the negative electrode to the positive electrode. When it moves according to the size. So as it moves, the larger fragments remain in the front, and the smaller fragments move faster, so they remain at the end. So they move according to the size. So the cutting of DNA by restriction into nucleus results in the fragments of DNA. So obviously, when you cut a DNA by restriction enzyme, it will lead to fragments of DNA because you're cutting them. These fragments can be separated by a technique. How to separate these strands? They can be separated by a technique called as gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards the anode under an electric field through a medium or matrix. Nowadays, the most commonly used matrix is agarose. So the gel here that we use is an agarose gel. Which is a natural polymer which is obtained from seaweeds. The DNA fragments separate according to their size. They are separate according to the size through sieving effect provided by the agarose gel. Hence, the smaller the fragment size, the farther it moves. The separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide. Ethidium bromide is carcinogenic. So we always handle it with the gloves. I've done this experiment, so I know. And you cannot actually visualize just like that. The agarose gel, the bands will not be visible to you in the through the naked eye. We have to you expose it to UV radiation, and then only we can actually visualize the bands. You can see bright orange colored bands of DNA in ethidium bromide stained gel exposed to the UV light. The separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as elution. So it can come from 1 mark or 2 mark. What is elution? The DNA fragments purified in this way or got this way are used in constructing DNA, recombinant DNA. So we can actually, the separated DNA fragments or desired DNA will be one of them, and we can actually use it to make recombinant DNA. So it's constructing recombinant DNA by joining it with the cloning vectors. So this is how it will actually be visualized in the UV. When it is actually exposed to UV radiation, these bands are visualized. As you can see, the fatter the bands are, they are actually at the they are near the well. They are near the well, and these bands which are actually thinner, they are actually towards the end of the uh, agarose gel. So the fatter the bands, they are actually towards the well, and the thinner and the smaller the band, they are actually towards the end of the gel. So here you can visualize it nicely. The largest bands will remain towards the well, and it is actually moving in this direction. And the smaller bands will actually be towards the end of the gel. Thank you for watching, and if you really liked my video, please like and share it with your friends. And if you have any doubts, please mention them in the comments down below. Thank you for watching.